41 is about the metabolism integration. So the three different types of metabolism that we read in this biochemistry course: carbohydrates, proteins, and lipids. How can we join these together, or where they are linked inside our body is going to be dealt in this lecture. So the learning outcomes are the tissue specific metabolism, what kind of metabolism happen in different tissues, and integration of the metabolic pathways, and how can we regulate these metabolism and metabolic pathways with the help of hormones. So this slide is about the division of labor of the tissues. Different tissues perform what kind of different functions in the body related with the metabolism. Liver mainly processes the nutrients or the diet that we eat. It is processed in the liver. Main function of adipose or fat tissue is to store and supply the fatty acids. So storage and supplying of fatty acid. And then we have a special type of adipose tissue known as brown adipose, which is thermogenic. Thermogenic means it keeps our body warm in cold weather. Muscles, as we all know, uses ATPs and do the mechanical work for us, any kind of movement. Brain uses energy for the transmission of electrical impulses, and it controls all other organs with the help of these electrical impulses. Blood is a transport medium basically inside the body, so it carries oxygen and metabolites and some hormones as well to different parts of the body or different organs of the body. So blood can be considered as an organ as well in the body, and it has got two main parts. One is the blood plasma, which is the liquid or the fluid part, and the cells that are present in it. And there are three types of cells: red blood cells, also called erythrocytes, which are in most abundant quantity; leukocytes, white blood cells, and the platelets, which are almost equal to each other, but most of the cells are red blood cells. Blood plasma is mainly composed of water, and then there are some solutes, some dissolved particles in that water as well, and these are metabolites, some waste products, oxygen, carbon dioxide, different hormones, carrier proteins. So all of these substances are basically suspended in the blood plasma. They are dissolved in the blood plasma, and the blood transports all of these substances. So pancreas is a very special organ, especially with respect to the hormones that it secretes. These hormones are very important for the body, and they play a very important role in the metabolism in handling our body. So there are three main types of cells. Alpha cells secrete glucagon, which increases the blood glucose levels. Beta cells secrete insulin, and there are gamma cells which secrete another hormone, somatostatin. So insulin and glucagon are the more important ones, you can say, whose function is totally opposite to each other. For insulin and glucagon, their function is totally opposite. Insulin decreases blood glucose and glucagon increases blood glucose, and these cells are found in a part of pancreas which is known as aisles of Langerhans. So aisles of Langerhans in the pancreas have got these different types of cells, and the pancreas has endocrine as well as exocrine function. Endocrine means different types of hormones, and exocrine means different digestive enzymes. So both of these functions are played by the pancreas. Here we have different properties and the functions of the liver. And the liver adapts to the changing metabolic conditions. What kind of metabolic conditions our body has will have direct effect on the functioning of the liver. So portal vein is the main vein of the liver that carries nutrients to the liver from a small intestine. The food that we eat is carried through the portal vein into the liver. Hepatocytes, which are the liver cells, turn these nutrients into fuel, convert them into energy, which can be used by all the cells of the body. And these hepatocytes enzyme, they have to function a lot, therefore they turn over quickly. They are quickly, their age is less, so when they get old they will be broken down and we will recycle them into new enzymes. And enzymes of these liver cells can increase or decrease with the changing diet and need of the other tissues. If you are eating too much or your diet contains specific food materials, this can increase or decrease the activity of these liver enzymes. Now the liver cells, which are the hepatocytes, have some unique features which are particular to the hepatocytes only. These are not found in any other cells of the body. And these are that the hepatocytes have GLUT2 transporters. These are special transporters for glucose movement in and out of the cell, and it is passive diffusion. And then hepatocytes have some special enzyme, glucokinase, which is the isotype 4 of the hexokinase enzyme. And this glucokinase has got a higher Km than other hexokinases which are found in other parts of the body, and it is not inhibited by glucose 6-phosphate. So these are key features of this glucokinase enzyme that is found only in the hepatocytes. And in the hepatocytes, fructose, galactose, as well as mannose are all converted into this G6P, which is known as glucose 6-phosphate, and this is known as a transfer station in the liver, because all of these monosaccharides, including glucose as well, will be converted into glucose 6-phosphate, and they will stop in the liver for some time, and then they can be sent anywhere else in the body. And this glucose 6-phosphate can be made by multiple pathways, and it can have multiple fates as well. Next we have the different types of metabolisms in the liver. A brief summary of the carbohydrate, protein, and lipid metabolism inside the liver. So for carbohydrate metabolism, glucose 6-phosphate or G6P is the starting point, you can say, or the central molecule of the whole metabolism. So it can have multiple fates, as we saw in the previous slide as well. If the cell wants to make energy, it can be converted into pyruvate and then later on go to the citric acid cycle and electron transport chain to give us ATPs and energy. If the liver wants this glucose to be sent to some other organ, it will remove the phosphate from it, convert it into free glucose, and it will go into the bloodstream, and from the blood it can be transported to various parts of the body. And if we have too much of extra glucose, then we can convert it into liver glycogen and store it in the form of glycogen to be used later on. If the cell wants to make nucleotides or DNA and RNA, this glucose can go in this pentose phosphate pathway and give us the sugar to produce these nucleotides. And when extra pyruvate is being converted into acetyl-CoA, this acetyl-CoA can be converted into the triacyl glycerols and phospholipids, so we can make some fats from it as well. Moving on to the amino acid metabolism, so the free amino acids that are present in the liver, they are mostly used to give us protein. So most of them are converted to proteins for the liver and other tissues. We can also make hormones and nucleotides from these amino acids, and also when they are broken down, they can be converted into CAC intermediates, which can be used to generate energy, especially for the liver. Moving on to the fats, fatty acids are the smallest particles which are usually present in the liver, and they can be used to make liver fats or liver lipids. And if the cell wants energy from them, we can convert them into acetyl-CoA. Same thing like this. a citric acid cycle will happen like it happened in the carbohydrate metabolism, and later from electron transport chain we can get energy from them. If acetyl-CoA is too much, it can be converted into cholesterol. From cholesterol we can synthesize steroids and other hormones like androgens and progesterones, and we can also make bile salts from them. And beta oxidation of fatty acid will give us plenty of energy as well. And these fatty acids can be taken into the bloodstream, and these fatty acids can be provided to other parts of the body as well. Here we have a comparison of the heart muscle cells and the skeletal muscle cells. So heart muscle cells are very active, as we all know that the heart has to continuously work. It keeps on pumping, and there is no stopping for these heart muscles as well. And for pumping or contraction, this heart muscle cell requires ATPs, and ATPs, as we know, are synthesized in mitochondria. Therefore, heart muscles have got more mitochondria than most of the cells of the body. 50% of the cell volume are mitochondria in heart muscles, and the primary source of energy for the heart muscles are fatty acids. Sometimes we can use ketone bodies and glucose as well as phosphocreatine for energy as well, but the main source of energy is fatty acid. And it's an aerobic organ. It always requires oxygen. If oxygen is cut off due to any reason, the muscle cell will die, and the disease that results is known as myocardial infarction, or simply in the layman's term we know it as heart attack if oxygen is not provided to the heart muscle cell. So here we have a little bit of explanation of the muscle contraction, uh, and how or which sources of energies will be providing this energy for the contraction. So normally, or you can say light activity or at rest when you are not doing any exercise, the fatty acids are the primary source. Sometimes ketone bodies as well as glucose are required, but as we saw in the previous slide, that fatty acid will provide most of the energy, and it will be converted into carbon dioxide, and it will be aerobic oxidation in the presence of oxygen. Now, if there is heavy activity or very strong exercise, then muscle glycogen will be utilized, and this will be anaerobic respiration or anaerobic metabolism, in which oxygen will not be there. Glycogen will be converted into lactate. Also, during heavy activity, we can use phosphocreatine as well, which is converted to creatine, and all of these substances are going to give us the ATP, or the energy for the muscle contraction. Here we have the repetition of the Cori cycle, which we have already discussed before as well. So during contraction or heavy exercise, glycogen is going to be broken down into lactate to give us the ATP or energy. This lactate can be taken into the liver through the blood, and in the liver we can convert the lactate back to glucose, and glucose then go back into the liver to provide more energy or more ATP. So it's a cycle between the muscles and the liver to continuously provide the muscles with the glucose, so that we can get the extra ATPs during the exercise. Next is the brain metabolism. So mammalian brain consumes about 20% of oxygen even at rest as well. So even when you are resting, your brain uses about 20% of all the oxygen in the body, and there are different types of cells present in the brain, and most of these are known as neurons. Glial cells are also present in some quantity, and the whole of the brain uses about 130 grams of glucose per day to be utilized for energy purposes. So when you are on the normal diet, Glucose will be the primary source of energy for the brain, converted into carbon dioxide. The same aerobic oxidation in the presence of oxygen. We get the ATPs, and brain uses these ATPs especially for the electrogenic transport of different neurotransmitters and different ions. Now, when you are starving, when you are not eating for a long period of time, brain can use ketone bodies for energy purposes as well. And the main ketone body used is beta hydroxybutyrate for energy. Now, there are special types of cells present in brain known as astrocytes, and these astrocytes can use fatty acids for energy as well. And as I said before as well, most of the energy is mainly used to maintain the electrical potential across the neurons or electrogenic transport of ions. Here we have the main function of the adipose tissue or the fat tissue, and the main functions are to carry out glycolysis through the oxidative phosphorylation. And these fat cells can convert acetyl-CoA into fatty acids as well. But in humans, the liver is preferred place for the fatty acid synthesis, not the adipose tissue, but they can be synthesized here. We can use these fatty acids to make triacylglycerols or THEs when we have extra fatty acids present. And when you are on starvation, then we can use these fatty acids as well, or release the fatty acids and give them to other tissues as well. Lipases are the group of enzymes that break down the THEs into fatty acids, and this whole process of THE breakdown is accelerated by the hormone epinephrine. And we discussed this epinephrine in detail in the signaling molecules or the lectures about the signaling molecules, in which we have the full cascade of CMP dependent reactions. And insulin decreases the activity of this enzyme HSL, hormone sensitive lipase. If you remember, hormone sensitive lipase was the main enzyme to break the THEs into the fatty acids. And the brown adipose tissue, its function is also important. It acts as a thermogenesis. It has a special protein, thermogenin, which keeps our body warm in cold weathers. Next is the fuel or the nutrient and energy use over a few hours or 4 hours of normal human metabolism. Immediately after the meal, when you have taken the food or meal, the glucose levels will increase in the blood, and it will secrete insulin. And insulin will stimulate glycolysis and glycogen synthesis, so that these glucose levels can be decreased back to normal levels. When 2 or more hours have been passed after eating, which is known as postprandial, the blood glucose will start to drop now. So when it drops below the normal levels, the opposite hormone glucagon is secreted, and its main function is to increase the blood glucose levels. So it will break down liver glycogen to give us glucose. And once 4 hours have been passed, when you last eat your food, more glucagon will be released, and it will also break down the THEs, and fatty acid will become the major fuel for muscle and for the liver as well. Here we have the general slide about the function or the control of insulin and glucagon on the glucose metabolism. So we just saw that as well. High glucose level stimulates insulin, and it will block the glucagon secretion, because their functions are totally opposite. Whereas the low glucose levels in the blood will stop insulin, and it will stimulate glucagon release, and glucose will enter the pancreatic beta cell via GLUT2 transporters and alpha cells through the GLUT1 transporters. These are the two different types of cells of the pancreas. Beta cells secrete insulin, and alpha cells secrete glucagon. So glucose enter in these cells through different transporters. So how does insulin decrease this blood sugar levels when you have high blood glucose levels? And its metabolic effects are increased glucose uptake by muscles and adipose tissue. So these cells take more glucose inside from the blood, and the enzyme is basically glucose transporter, which will be more active and takes more glucose in. High glucose uptake by the liver as well, which is through the glucokinase enzyme. Increased synthesis of glycogen. Glycogen is the polymer of glucose. If we have extra glucose, we can take it from the blood inside the cells and we can store it in the form of glycogen. Whereas the opposite process will be stopped. So glycogen breakdown is down regulated or is inhibited. The main enzyme for glycogen breakdown is glycogen phosphorylase. This enzyme is inactivated by insulin. Glycolysis will be increased, which gives us more energy. Fatty acid synthesis will be increased through the ACC enzyme, and TAG synthesis will also be increased through the lipoprotein lipase enzyme. So all of these metabolic effects are basically to decrease the blood sugar levels through the hormone insulin. So in the well-fed lipogenic liver, well-fed means when you are eating a normal and regular diet at regular intervals. The pancreas will secrete insulin, and insulin has the effects on the liver that we saw, especially on the glucose metabolism. These are the ones that we saw previously as well. Glucose mostly will be converted to glycogen to be stored, can be converted into acetyl-CoAs as well, and we can synthesize the TAGs or the triacylglycerols, which will go to the adipose tissue and be stored there. And the extra glucose can be sent to a brain as well, which can use glucose for energy purposes as well. Whereas the amino acids in the well-fed state are mostly going to be catabolized and sometimes used for protein synthesis as well. The remaining will be catabolized and converted into urea and give us energy as well. Whereas the fats again will be converted into the TAGs and be stored in the adipose tissues. The opposite hormone of insulin is glucagon, whose main function is to increase the blood glucose level. So this hormone is released when you are starving, not eaten for some time. Then your blood glucose levels are falling, and the main function will be to increase the blood glucose levels. So all the metabolic pathways on which it will occur, the main focus is to increase the blood glucose levels. So it will break down glycogen more. Glycogen breakdown will be increased, and therefore when we break the glycogen, we get more glucose that can go into the blood, and the enzyme it works on is glycogen phosphorylase. The opposite pathway, glycogen synthesis, will be decreased, and this enzyme glycogen synthase will be inhibited. Glycolysis will be decreased as well, especially in the liver, because we have to be careful in using the glucose. We can't be making too much energy from the glucose available. It has to be you can say saved for the important organs like brain. So the enzyme PFK1, which is the main enzyme of glycolysis, is inhibited. Increased gluconeogenesis so that we can get more glucose in the blood. Increased fatty acid mobilization or breakdown of fats, and increased ketogenesis as well. All are functions of this glucagon to increase the blood glucose levels. Now the fasting glucogenic liver will be when you are fasting and not eaten for some time, and the main hormone acting on liver in this case will be glucagon. So as we saw in the previous slide as well, glycogen will be broken down into glucose molecules from the amino acids, and from the pyruvate as well. We will be making more and more ketone bodies to be supplying the brain mainly. First up, it will use the glucose, but when glucose is almost finished, then the brain can use the ketone bodies for energy as well. Also, we will be breaking TAGs a lot, and they get the glycerol to be converted into the to be used in glycolysis, as well as the fatty acids that we get from breaking down TAGs. They can be used in making the ketone bodies, and ketone bodies can also be used by the muscles for energy purposes as well. Next, we have effects of prolonged fasting. Prolonged fasting means when you are fasting for many hours, more than 10 hours, 15 hours, and can be over the span of days as well. So when you have not eaten for a long period of time, the muscles will begin to be used for fuel purposes. What this means is you will break down your muscle proteins, convert them into amino acids, and use those amino acids for energy. The liver will deaminate, take the nitrogen out, or transaminate these amino acids as well, and the nitrogen or the amino groups will be converted into urea. The carbon skeleton can be, which especially the ones that are glucogenic amino acids, they can be converted to pyruvate, and then to finally glucose, and we can use that glucose for energy. And this glucose will mainly be provided for the brain, because in such emergency situations, brain is the most important organ that must be provided with energy. The fatty acids will be oxidized to acetyl-CoA, so we will break down our fats into fatty acids, and those fatty acids will be converted into ketone bodies, and ketone bodies can be used for energy purposes by many tissues. This ketone body synthesis happens in the liver. So explanation of the previous slide, effects of prolonged fasting in the form of diagram is presented in this slide. And prolonged fasting is a situation which is similar to diabetes type 1 as well. In diabetes type 1, our body behaves in the same way as, especially the liver behaves in the same way as it would in prolonged fasting. So as we saw in the previous slide, proteins will be broken down to give us amino acids converted into glucose, and glucose can be used for energy purposes, and the fats will be broken down as well into fatty acids. Fatty acids will mainly be converted to acetyl-CoA and eventually into the ketone bodies. Again, these ketone bodies as well as the glucose will be provided to the brain for energy purposes as well. And if the glucose is completely lost for glucose, there is no glucose present, and glucose can use these ketone bodies for energy purposes as well. So here we have a graph like the presentation of the days of starvation and the plasma concentration of some of the common molecules that are found inside the body. And as we can see, when the days are continuously increasing from 0 up till 40, the ketone bodies with the most common one, beta hydroxybutyrate, it keeps on continuously increasing and increasing. Glucose levels slowly keeps on falling over fasting. Free fatty acids, they rise up very quickly in the first couple of days, then almost stabilizes. Same is the case with acetoacetate and acetone. Gas is also going to be continuously increasing as well. Last up, we have the role of epinephrine, which is a hormone which has very similar effects to glucagon that we just studied earlier, and opposite effects of insulin as well. So first up are physiological effects of epinephrine, which is also commonly known as adrenaline. And the main effects it has is it increases the heart rate, increases the blood pressure, and increases the dilation of the respiratory passages, so that all of these three effects are to increase the delivery of oxygen to different tissues, especially the muscles. And the metabolic effects are the same as glucagon. It increases glycogen breakdown, decreases glycogen synthesis, increases gluconeogenesis, increases glycolysis, increases fat breakdown or fatty acid mobilization, and it also enhances glucagon secretion and stops insulin secretion. So most or all of these metabolic effects are mainly to produce more glucose as this hormone is released in situations when the body is low on glucose or the body requires more glucose than usual.